Hello and welcome to Stars, Cells, and God. My name is Hugh Ross, and today I'm joined by Fazale Rana, our staff biochemist, and also the president of Reasons to Believe. And I'm going to be talking about axions, how that pertains to dark matter, and how that ties into the cosmic creation model. And Fuzz, you're going to be talking about how to store all my data on DNA. Well, welcome, and we're happy to have you here.
Yeah.
Fuzz, why don't you take it away?
Yeah, sure. And just to have a little bit of fun before we get into some heady stuff, I'm just curious if you know what this is a picture of.
Well, it looks awfully black, and maybe I can see Venus and Jupiter up at the top, but I suspect this is not astronomical.
No, it's not.
Because I see that little paperclip thing there just
Okay.
off center.
Well, that's actually a blue D.
As a blue D, okay.
And this was the Hollywood sign after the Dodgers won the World Series. They turned all the lights off and they lit up the D to be blue in Dodger blue.
I missed that.
Yeah, so this is a little fun thing they did to celebrate the Dodgers World Series victory. But here's actually the Hollywood sign in daylight. And this is, of course, an iconic image that everybody is familiar with. People come from all over the world to see the Hollywood sign, and you know that you're in Hollywood when you see the sign. But this type of signage actually points to something rather profound, and that is that we recognize whenever we see information that there is an intelligent agent, human beings that are responsible for generating that information with the idea that they're trying to communicate something. In this case, that you are in Hollywood, right?
So you're telling me that the Santa Ana winds didn't just randomly put all this stuff up?
No, they did not. And that's, kind of the idea is that our common experience is that whenever we encounter information of any type, we know that there is somebody trying to communicate information, to somebody else, that there is a message that's being communicated. And that really forms the basis for one of the more popular and I think compelling design arguments that Christian apologists make using biochemical systems, noting that biochemical systems are information systems. And again, information comes from a mind, so therefore, if biochemical systems harbor information, then they too must come from a mind. So the idea here is that the information content of biochemical systems is evidence that there is a creator that's responsible for the origin and minimally the basic fundamental design of biochemical systems and life. And I think most people are familiar with the structure of DNA. DNA is the quintessential example of a information harboring biochemical system. In fact, the primary role of DNA in the cell is to store information that the cell's machinery needs to make proteins.
And to do it in a very stable way.
Yes. And in fact, DNA not only stores the information needed to make proteins, but also information that regulates how much particular proteins are which proteins are produced and how much of those proteins at particular points in the cell cycle or at particular points during the process of embryonic growth and development. And the information in DNA is digital information. It's also analog information, but that's a whole nother complex conversation that we're just going to put on the shelf. But the way to think about it, it's digital information, right? And digital information consists of discrete elements that are sequenced in a particular way, where that sequence of those elements is harboring information. So in a language, the letters of the alphabet are combined in particular ways to form words which communicate, distinct information, where different letter combinations form different words. Same thing with DNA, where different combinations of the subunits of DNA, which are called the genetic letters or an abbreviated A, G, and C and T, are like the letters that are used to make words.
So it's a four-letter alphabet.
Yes, it's a four-letter alphabet that basically is specifying, again, different types of proteins depending on the nucleotide sequence, right? So That's one way in which information is stored in DNA is in the nucleotide sequences. Another way is through what are called epigenetic modifications of the DNA. So one way in which an additional layer of information is stored in DNA is to methylate the cytosine residues, which are abbreviated as
Tell
C.
us what methylate means.
It means to simply attach a methyl group to the cytosine molecule. In fact, this slide shows a reaction where cytosine is methylated using an enzyme called DNA methyltransferase. And there's a number of different DNA methyltransferases that recognize specific sequences in the DNA and then will attach a methyl group to cytosine in those particular sequences. And again, there's a number of different DNA methylases that are involved in these methylation reactions. Now, this methylation actually is a critical way of regulating which types of proteins are available for the cell to use. So during the course of embryonic growth and development, where you have in the early embryo these cells that are highly generalized, that over time become specialized into the 210 different cell types that are part of the human body, there are these epigenetic changes that are taking place, modifications of the DNA that are permanently turning on or turning off genes. And this is a way in which gene expression is controlled and cells specialize. Or you can have epigenetic changes in response to environmental conditions. So, for example, if people are starving and they're in a famine and there's not enough food, there will be epigenetic changes that take place in their genome so that they hoard calories, right? So these are but you can think of this as an additional layer of information that's sitting on top of the nucleotide sequences of DNA. So the idea here would be, look, we've got multidimensional information now associated with DNA in the nucleotide sequences and in the methylation pattern, which could be thought of as being a type of or a set of instructions that is telling the cell's machinery which genes they can access, which genes it can't access. So this is, part of the argument for design, right? Information comes from a mind. This looks to be examples of two sets of digital information systems that are, part of DNA. Now, the response that skeptics bring to that particular design argument is illustrated by an article written a few years ago by Massimo Pigliucci.
Mm-hmm.
He's a well-known philosopher of biology and a critic of intelligent design. And he wrote an article where he makes the point that when biochemists talk about information, quote unquote, in biochemistry, they really are not talking about true information. It's more a metaphor that is convenient to essentially give us a perspective on the chemical structures and the chemical processes happening in the cell, that it really is wrong to think of this as being genuine or bona fide information. And so this is a criticism that really has its origins in the work of David Hume, who criticized a version of the design argument called the watchmaker argument, which is based on analogical reasoning, which is how this design argument works as well. Look, information comes from a mind. That's our common experience. We see information in the cell, so by analogy, we assume that information too came from a mind. But what Pigliucci is pointing out is that there's a difference between the information we create as human beings and what we actually see in the cell.
Sounds like he's using the same approach as David Hume, basically saying the analogy is not tight enough.
Yes, exactly. Yeah. Right. And that this is, that we want to be very careful about drawing, these kind of watchmaker conclusions because of it's really not information. And this is where the whole idea of DNA data storage comes into play, because there's a whole area of nanotechnology where people are looking at using DNA as a medium for data storage. And this is being driven by the fact that we're facing a data storage crisis.
Yes, we are.
You astronomers and we biochemists are generating vast amounts of data.
Not to mention all the cat videos you see online.
On YouTube. And so, there are projections that within a decade or so, we may have created so much digital information that we literally won't have the means to store that information anymore. Through the use of magnetic tapes and things like CDs and hard drives. We just simply won't be able to store that data. And that's going to lead to some very difficult decisions as to what data makes the cut.
And what data doesn't, right?
Right. And so people are looking for ways to come up with alternatives. And one idea that's gaining quite a bit of momentum is to use DNA as a data storage medium. And we don't have the time to do this here, but in my book, Fit for a Purpose, I talk about how all the different structural features of DNA are highly optimized for it to function as a data storage system. And that there are some people who think that DNA may actually be at the theoretical maximum for the amount of information that can be stored per unit mass.
And per unit volume, too.
Yes. Yeah. And so it's an incredibly efficient data storage medium, which, makes sense when you think about how much information it's storing in order to direct the operations that are needed for the cell. But just a statistic I've seen is that 1 kilogram of DNA, which is about 2.25 pounds, can store all the digital data that exists in the world today.
Right.
That's, that gives you a sense for the data storage capacity. And this is an older study now, but George Church and his team at Harvard University in 2012 demonstrated as a proof of principle that DNA could actually store information. And the way they did this is by literally encoding a book, which was a 55,000 word book with 11 figures. They encoded that into DNA. And it's a very clever set of tricks they used to be able to pull out individual pages, so that you could not only encode the information, but then retrieve the information. Now, with that particular study, what they did is they decimated Massimo Pigliucci's argument, because the way in which that data is being encoded in DNA is identical to how information is stored in DNA to specify the production of proteins. Right. The only difference is, in one case, you're specifying the production of a page, in a book. In the other case, you're specifying the production of a molecule. But either way, you are storing information in DNA and encoding it precisely the way that the cell's machinery encodes it.
Well, it's also a very tight analogy to how we store data on a flash drive.
Mm-hmm. Yes, exactly. So this, again, as I said, is a really strong response to Massimo Pigliucci's challenge. The problem with this approach is that it's very cumbersome. DNA is optimized to store data and for the cell's machinery to retrieve data very quickly in a biochemical context. But when we're now using it in a technology context, it's a very cumbersome way. Even though it has an incredible data storage capacity, it's very cumbersome to write the data to DNA and to retrieve the data from DNA. So that means it's really only going to be useful for long-term data storage where you're not looking to access that information. And part of the problem is, in order to store the information, you literally have to synthesize DNA according to particular sequences.
Right.
Now, there are automated synthesizers that can do this, but the problem is that those synthesizers can only produce reliably DNA sequences of about 200 genetic letters. Much beyond that, you start introducing errors. And even though these DNA synthesizers have a high degree of accuracy, even a very small error, once you start adding 200, genetic letters starts amplifying so that you reach a point where you're going to introduce significant number of errors as the sequences get larger and larger. So what you have to do is not only is sequence the DNA, synthesize the DNA, but then you have to catenate it together using enzymatic techniques. So it's very cumbersome. So the paper that I wanted to talk about was published by a team of Chinese researchers, and this is a picture of the paper, where they take advantage of the idea of epigenetic modifications to store the data as opposed to using the DNA sequences. And I'm going to geek out for a little bit and just talk about the work that they did, just because it's so clever. So it doesn't really have any contribution to the overall apologetics point, but it's very clever.
Well, let me just stop you there. I mean, it sounds like this paper's found a faster way to get the data on the DNA, and probably a faster way to recover it.
Yes.
So that's the bottom line?
That's the bottom line.
Yeah.
But it's a very clever approach. And so again, what they're using, the DNA double helix is actually functioning as it's not the template, it's actually the storage vehicle, right? Whereas the information is actually in the DNA methylation or in the epigenetic changes. And so this diagram shows you have the DNA template, and then the next layer up is actually the information layer, where if there's a methyl group, that's a 1. If there's no methyl group, that's a 0, right?
So it's just like our computers.
Right.
Yeah.
And so what they do is they construct the double helix by having a single strand of DNA, and then they have these smaller pieces of DNA that will pair in precise locations along that DNA single strand. And they can either methylate those really short pieces of DNA or not, and then they expose those short fragments to the single strand, and it forms a double helix where there's methylation patterns. And then they do something clever where they then use an enzymatic trick to transfer the methylation from those short fragments to the single strand, right? So now you've got a permanent single strand of DNA with a methylation pattern, and that methylation pattern now is storing information where, again, a methyl group's presence is 1, absence is 0. Then you can do interesting things, like here's an example of if you had an image like a panda or this Chinese image of a rubbing tiger, you can create a matrix, right? And then what they've done is they attach to that matrix a matrix of 1s and 0s, which is depicting the information needed to reconstruct the image. They append to that a barcode, right? Then that barcode tells you where you're at in the matrix. So then what they can do is they can do something that's analogous to typesetting, where they have a tray that is a set of wells, and in those wells you either put the enzyme to methylate DNA or you don't. Then you add the short fragments, and then the methylation reaction will happen or it won't. And then you can expose those fragments to that single strand of DNA, right? And each of those strands has a region that is like a barcode that allows you then, in effect, to reconstruct a matrix, right? That you can then read out very quickly through DNA sequencing, right? So where you use that barcode and the methylation patterns after you sequence it to determine what the matrix looks like. And as a way to show that this is actually a viable technology, they had about 100 volunteers who had no experience working with DNA actually encode information, showing that they showed them how to do it, and they were able to do it pretty quickly, in a non-laboratory setting. And so this is one of the authors' point, is that desktop DNA printers or storage kits could be developed for use at home or in small organizations, enabling users to back up important personal data, such as legal documents or digital photos, in a form that can last for centuries. So we're seeing, with these kind of ideas, this conceptual idea of DNA data storage being something that very well could become part of our lives in the near future. So this is really pretty exciting stuff.
Anything in the paper about the timing? I mean, for example, if I wanted to take, say, a page out of my passport and get it into DNA storage, how long would it take a volunteer to do that?
I don't know the answer to that question off the top of my head. I didn't pay attention to that in the paper.
But less than a day?
Oh yeah, I think it may be an hour or so.
So less than an hour. Well, that's impressive.
Yeah. So
Okay.
now, I mean, what they were doing is they were encoding fairly simple messages,
Yes.
right? But just as to show that this technology is user friendly. To probably encode something like a page from the passport where you had a picture and text, that may be a little bit more involved, right? But the point is that if that's something that you want to store for long term,
Right.
then,
And you're only going to be accessing it maybe once a decade.
yeah,
So in which case, taking an hour to pull it out,
yeah,
putting an hour to put it in, that's realistic.
yeah, right. And it's just simply noting what's the sequence of ones and zeros, and then
Right.
just adding or not adding things to trays and wells. And it might even be something that could be automated, fairly quickly.
Yeah, I could see the potential to drive it down to a few minutes.
Yeah.
So,
But the point here is that, just like the early work in DNA data storage, driving the point home that the sequence of nucleotides is truly information. It's not a metaphor. We can also now make a corresponding point that the methylation pattern is not just simply a metaphor for information. It's actually information because these researchers are using it in that way. And the inspiration for this idea comes from how biochemical systems work. And so what they're doing is identical conceptually to what is happening in the cell in terms of creating that additional layer of information in the methylation patterns. So the bottom line is that this work that's happening in DNA data storage really drives home the point that what we see in the cell is information. And regardless of what philosophers of biology complain, it really is information. But we also can get a sense for just how sophisticated that information is when we start looking at the ingenuity that it takes these researchers to figure out how to use that information to encode the types of digital data that we manipulate and handle all the time.
So the criticism that the analogy isn't very tight, that criticism is gone now.
Yes.
This is an extremely tight analogy.
Yep.
So,
Yeah.
but also means we can look forward to archiving huge amounts of data.
Mm-hmm.
It's data you don't need to access very often. And, just getting that off the scientific table,
Yeah.
taking all the data in the world that needs to be archived, that's not going to be accessed on a daily basis, that would release a whole lot of computer capability
Yep.
for things like astrophysics and biochemistry, right?
Yeah, that's right.
So, yeah.
All right, Hugh, it's up to you now.
Okay, well, axions, they're probably the most sought after particle in all of physics and astronomy because, we know that the universe is comprised mostly of dark energy and about 30% of it is matter, but the matter comes in two forms, ordinary matter and dark matter. And detecting the ordinary matter is easy to do because ordinary matter particles strongly interact with photons, and photons are easy to detect. The problem with the dark matter is they're made up of particles that either don't interact with photons at all or interact at an extremely weak level, so they're difficult to detect. And really the only ones we've really detected so far are neutrinos. And we know that neutrinos make up only a very tiny percentage of the dark matter. And so both astronomers and physicists said, it's great that we can prove that dark matter exists, that it makes up about 23% of all the stuff of the universe. We know there's about 5.5 times as much dark matter as ordinary matter. We can detect its gravitational influences. But we do get a lot of skeptics saying, hey, Are we really certain about the Big Bang creation model? Because the Big Bang creation model fundamentally depends on the existence of dark matter and its existence at a level of about 23% of all the stuff in the universe. And if you guys can't even find the particles, how are we going to believe that this is really accurate? How are we going to believe the theological implications that a God beyond space and time must have created the universe and personally designed it so we humans can live? So being able to determine not only the gravitational influences of dark matter, but the particles that comprise it would be a huge step in advancing the creation model we have here at Reasons to Believe, the model that's embedded in what's called the Big Bang creation models. And, personally, I'm a little bit annoyed at this debate. It's like we don't have to find the particles to know that this stuff is real. And we know that it's real, and we know how much of it there is in the universe. That is enough to prove that the universe indeed did arise from a Big Bang creation event where you got a crater beyond space and time that created everything. But it would certainly be icing on the cake if we can find these particles. And, I've been writing for the past several years about the best attempts to find these particles. Maybe the stars that are orbiting the supermassive black hole, the center of our galaxy. Because wherever you got a lot of mass, and particularly a high density, high gravitational fields, those gravitational fields will attract the dark matter particles. And you need a high density of these dark matter particles to have any hope of detecting them. And so this paper was published in Physical Review X. Physical Review's got a whole lot of journals. Probably it's the most prestigious physics journal that exists in the world. Physical Review, A, B, C, and D, Physical Review Letters, and recently they launched Physical Review X, which is basically for the more speculative stuff and also the stuff that could significantly advance our understanding of physics. Those are the papers that get published in Physical Review X. And this one's titled Axion Clouds Around Neutron Stars. And the four physicists here are basically arguing, hey, the best place to try to detect these elusive particles would be around neutron stars. Neutron stars are extremely dense. To give you an idea, it's about 2 billion tons per level teaspoonful. So that's, quite dense. So, and these neutron stars are about 10 kilometers across, so you got a lot of mass, extreme density, very powerful gravitational fields. But the other thing they were noting is these neutron stars that are spinning rapidly, the pulsars, because, as they collapse, they will spin faster and faster. It's a conservation of angular momentum. What you see with figure skaters, they will start off like this and begin to spin, then they pull their arms in, and the conservation of angular momentum makes them spin really rapidly. Well, likewise, when a star like our sun, that's nearly 1 million miles in diameter, were to collapse down to, say, a neutron star that's 10 kilometers in diameter, you get an extremely fast spin-up. Some of them actually spin so rapidly, we see a pulse every few milliseconds. So it's literally spinning at close to the velocity of light. And so that will generate a powerful magnetic field. We're talking magnetic fields with some neutron stars that are trillions of times greater than the magnetic field of the Earth. So think of a magnetic field that's trillions and trillions of Gauss. And so what they're basically pointing out is that these neutron stars are going to collect a lot of these dark matter particles. And the assumption is that axions are probably the best candidate to make up dark matter, and they're way more numerous than the neutrinos.
Are they charged?
No, and their masses are really low.
Okay.
So yeah, if they were charged, then it'd be easy to detect and that's one of the properties of ordinary matter.
Oh, is it? Okay.
Yeah. So these dark matter particles are not charged, so that's why they're difficult. They don't interact with photons hardly at all, which makes them difficult to detect. These axions, though, are theorized under certain conditions to decay into photons.
Okay.
But the decay rate is extremely low, so you're not going to get much. And in order to detect the photons, you're going to need an extremely high density of these axions. But they were saying, okay, we all also know they're extremely low mass, and I tell you how low mass they are. They're thinking the most likely range is 10 to the minus 4 to 10 to the minus 9 electron volts. So incredibly low mass, a billionth of an electron volt, which means they're going to be extremely difficult to detect. But they're also showing that under certain conditions, these neutron stars are able to collect 10 to the 50th axions per minute. That's a lot of axions.
So how are they collecting it? Is it just gravitational attraction?
It's the gravitational attraction that collects them. And so what they're hypothesizing is that there's going to be these little clouds of axions surrounding the neutron star. Neutron star is basically a 10-kilometer ball of neutrons touching one another. So just have a whole bunch of neutrons packed so tightly together, literally touching one another, but there would be a cloud around this ball of neutrons that would be made up of axions. And as the axions escape from the neutron star, they're having to pass through a magnetic field that's trillions of Gauss. And as they rapidly move through this powerful magnetic field, you're going to get some of those axions decaying into photon pairs, which means we should be able to see a little bit of light. And so what they're proposing is that radio astronomers use their radio telescopes, their biggest radio telescopes, to see if they can see a little bit of enhanced light. Because we know how bright neutron stars should shine based on ordinary matter. If we see some additional luminosity, that would be a detection of the axions. So they said 2 ways we can detect these axions is use very broadband receivers. So you're basically looking over a huge part of the radio electromagnetic spectrum, which means you're going to accumulate a lot of light, radio light.
So they don't know how much energy these photons would have then as they are produced from axion decay. Is that the idea?
Well, they will if they can figure out the mass.
Okay.
And that's the other thing they're pointing out is that, we're looking over this mass range, and you're going to get a sharp spectral radio line at the mass of the axion.
Oh, I see.
So this has the potential to determine the mass of the axion, which will actually give you a lot of information on the decay mechanism, how the axions decay into photon pairs. But they're basically saying there's 2 ways that we can detect these axions. Number one, to see in a very broadband spectrum additional light, extra light, that would be in addition to what you would get from the ordinary matter. That'd be one way. The other way is seeing a sharp, bright spectral line that would be at the mass of the axion. And they said, we have the technology today to put this to use. So they basically end their paper saying, let's use the biggest radio telescopes we have with the broadest band receivers. Let's start looking at these pulsars and see if we can see some extra broadband light, or let's see if we can find a spectral line. It'd be neat if we could find both. Finding both would give you the most information about the axion. And they're saying that the radio telescopes that exist today have the capability of detecting these axions. Worst case scenario, the coupling between the axions and the photons is at such a weak level, all you get is an upper limit. But they said radio telescopes today can establish an upper limit on the coupling between the axions and the photons. That's 100 times lower than what we've been able to do so far. So that in itself would give us a lot of good information. So even a non-detection tells us a lot. And they said there's no other way we could get such an extremely low limit on the coupling relationships between, and basically we're talking about the decay. Because the only way you're going to detect these axions is if they decay into particles that we can see, namely the photons. So And already we got some quite low limits. We already know that the decay is going to be extremely difficult to detect, but here's a potential to go a factor of 100 down. And the real hope is, hey, if the coupling is strong enough, we're actually going to detect this additional light, see the spectral line, and then we'll actually know what the population of axions is in the universe, what their mass is, what the decay mechanism is for these particles to decay into photons. And that would be, quote, the holy grail of astrophysics, to actually come up with this information. So the paper is basically saying, we think pulsars is our best shot at finding these things. And of course, we can build radio telescopes that would specialize in the detection of this thing. So far, we can use existing instruments to establish a limit that's a factor of 100 times lower than we've been done so far. But we could actually go to the taxpayers and say, hey, we'd like to build a specialized radio telescope that'll, say, be able to establish a limit 10,000 times lower. And hey, they're basically saying if we can go that, we're going to detect these things. So,
yeah. Is there any thought that there might be other particles beyond axions and neutrinos as part of
yes,
dark
there's
matter?
about 35 particles that are in contention to make up dark matter. But most astronomers and physicists are persuaded that there could be several particles that comprise, we already know neutrinos is one of them, and we detected those.
Right.
But they make up an extremely tiny percentage of the dark matter that's out there. And so axions are the leading candidate. Some think axions will make up over 90% of all the dark matter out there. Others are saying, well, maybe it's just 50 or 60%. So, and maybe it's not a factor at all. Some other particles might be in there. But there's already some clues that axions seem to be a good candidate, because as we look at white dwarf stars, they seem to be cooling at a rate that indicates, hey, it's not just thermodynamic cooling. There seems to be some additional decay there that we're seeing in that. And so they looked at one white dwarf binary star and said, we're actually seeing something that's in addition to just pure thermodynamic decay. We think it's axions. Well, to really nail it down, you need to be looking at thousands of white dwarf binary stars. And one I wrote about, in fact, we did a news of the day back in July, and it's archived at reasons.org on our YouTube channel, where we talked about stars that are orbiting the supermassive black hole in the center of our galaxy. And these are very high mass stars. But as we look at the spectra of these stars, we see that they're too old. And so the thinking is, if they're only looking at ordinary matter, they should have burnt out by now. And therefore the thinking, what's causing these really big stars orbiting a supermassive black hole to continue showing off light? There's additional light coming from axions. And so that's a project underway. So actually what I'm trying to share with you is that there's actually multiple astronomical efforts to try to detect these. And so they already have some promising evidence. We're seeing these Methuselah stars at the center of our galaxy, big bright stars that are too old to be only showing those characteristics if it's only ordinary matter that's responsible for the light. And then also seeing these white dwarf binary stars, but none of them are at a level to win the Nobel Prize.
Right.
We're looking at maybe 2 or 3 sigma above the background noise. I think in the case of white dwarf binary stars, it's close to 4 sigma. But the standard is you got to be at 5 sigma, 5 standard deviations away above the noise in order to claim I got a real detection. And you obviously want to have a few examples where you're above 5 standard deviations. So, but the fact that we're at 3 and 4 already tells us this is looking promising. And so this group is saying, we think we can do better by going after these pulsars.
What are the differences in the properties of neutrinos and axions? And does that difference contribute to the behavior of dark matter?
Well, the big difference is that the neutrinos are at a much higher mass level,
Okay.
which is why they're easier to detect. So these being very low mass. And so people are saying, hey, if they're between 10 to the minus 4 and 10 to the minus 9 electron volts, we got a shot at detecting them. If they're like 10 to the minus 15 electron volts, forget it.
Yeah.
So,
Yeah. So then this is adding to the case that there is a cosmic beginning, right? Then, and if there's a beginning, there must be a
well, as I
beginning.
said, it'd be icing on the cake. I think we already got more than a strong enough case that the Big Bang creation model, which is fundamentally dependent on the existence of dark energy and dark matter, and people have been saying, well, we doubt both, because with dark energy, we've yet to identify the constants that are responsible for the dark energy. And like with the dark matter, we haven't found the particles that make up most of dark matter yet. So these are outlying problems with both dark matter and dark energy. But as an astrophysicist, as I'm saying, I don't see any of my peers doubting that dark energy exists or that dark matter exists. We see its impact on the geometry of the universe. We see its impact on the cosmic expansion rate. But it'd be really nice if we can establish which constants are governing dark energy and which particles actually comprise dark matter. And hey, if we can find these particles, that's really going to shore up our particle creation model. And one of the things I think ways we can promote what the Bible says about creation is that we would expect the particle creation model to be compatible with the cosmic creation model, that they're both saying the same thing. And what we discovered so far shows that that is true. But again, it would be really nice if we could actually demonstrate that more comprehensively than we have so far. So that's basically what's going on in this research. As an astronomer, I'm excited because I think there's real potential just to use even existing instruments to actually have a shot at, quote, winning the Nobel Prize by finding these particles. And it will win the Nobel Prize. Whoever finds these axions beyond any reasonable shadow of doubt will win the Nobel Prize.
Right. Well, I mean, it's revealing something fundamental about the nature of the universe. That's Nobel Prize worthy.
Nobel Prize worthy, but it's also showing why you got these different observing groups using different ways to detect these things. My concern is somebody might jump the gun and say, hey, you know, we found it, but then they do the data analysis and say, well, we're not quite there. You really want to be certain that you found it. As I said, one white dwarf binary star won't do it. One Methuselah star in the center of our galaxy won't do it. You need a lot of observations. And likewise, you don't want just one pulsar. You want thousands of pulsars showing the effect. So that's it.
Yeah, interesting stuff.
It is. Well, it's always fun, Fuzz, to talk to you about these discoveries. And hey, I just want to share with all of you out there, too. Thank you for joining us today on Stars, Cells, and God. Join the discussion in the comments below. We actually read those comments. Remember to like this video and to subscribe for more content on the Reasons to Believe YouTube channel. New episodes of Stars, Cells, and God release each Wednesday and are available here on YouTube and on your favorite podcast app. Be sure to share this video with a friend, and remember, the more we learn about science, the more reasons we have to believe in Jesus Christ as Creator, Lord, and Savior.